Attempt to try to get through it. So yeah, boss, thanks for letting me talk. I should say that this talk is actually about my previous life in oncology, not on Alzheimer's, but basically all the principles sort of the same. So I'm a medicinal chemist. I'm not a pharmacokineticist, and I'm not a modeler. So sort of this idea really is to just give a very brief overview, if the slide would move, as to how we use PKPD in both drug design, picking clinical candidates, and then moving a compound into the more advanced stages before you get into preclinical and then to clinical studies. So just breaking it down to those two parts. So the PK part is pharmacokinetics. If you're not hugely, or if you haven't seen much of pharmacokinetics before, it's quite a long and complicated subject, but this is basically what you get to see. And all pharmacokinetics really is the changing concentration of your drug over time. And typically you take somewhere you can sample, which is almost always the blood or the plasma, and you see how your compound concentration changes. This particular one, PK studies, you see in the bottom left-hand corner. So this is actually of our clinical candidate, and it was the low-dose mouse PK that we used to then work out how the drug was going to work or whether the drug could work and how we would dose it in order to show efficacy and PD modulation. So we always take an IV dose as well as an oral dose. The reason for this is sort of how the theory works. If you don't have an IV dose, you can't work out some things. But the most important part is most of the time we're not hugely interested about what's going on in the plasma. It's just somewhere that we can sample quite easily, and it's rarely the place where our site of action is. And we want to find out what's going on to our drug when it's in all the different places within the body, particularly at the site of action where our protein target is in order to show the effect. Now, to do that, we apply quite a lot of theory, and what you get back from your friendly PK team is this vast number of collection of numbers that you see in this box above here. All of these numbers are derived from this PK curve and just using sort of theory and ideas to what they are. But we need to understand what each of them sort of do. So this is one here called AUC, which is the exposure of the drug, which is super important. Clearance is one you hear about quite a lot, and this is how the drug is left, gets out of the body. Half-life is how long it's in there for, and there's really unusual term called volume of distribution, which really messes with people, but it sort of tells you where your compound is sticking to when it goes to different places. And at the far end of this sort of more famous one, oral bioavailability. Now, you might collect all these numbers together, but to be honest, they don't really tell you a huge amount, and they don't tell you a huge amount about whether your drug could possibly work. And the reason for that is this big idea called the three-drug theory. So even though you almost always just measure the concentration of your drug wherever you sort of can, that isn't really what's useful or what's driving the pharmacological effect. That has to be driven by the free concentration. Now, the simplest model we could sort of have of this is having two compartments, compartment A and compartment B. And if you have a semi-permeable membrane down the middle, you'll see in our little blue blobs here is where they could be proteins or just anything really for your compound to stick to. Now, when your compound is stuck to those blue blobs, it's out of the game. It doesn't drive pharmacology. It can't be cleared from the body. It just acts like a reservoir floating around inside the animal and eventually inside the human patient. Now, because of the semi-permeable membrane, the blue blobs, because they're nice and big, they can't go in and out or go through that semi-permeable membrane, but our drug molecule can do. So you will find that the total drug concentration on either side of that semi-permeable membrane can be different, and almost always is very, very different. But it's the free concentration on the either side of that semi-permeable membrane will have to be the same when the system is at equilibrium. And we use this idea to really drive how we interpret PK and then how we extrapolate that to PKPD and understand the effect of our drug and how it's working. So it's a super, super important idea. Now, once we pull those two ideas together, we can understand the free concentration of our drug. We can understand the free concentration of our drug somewhat at the site of action, as long as you apply the theory properly. And then to pull in the PD part, we then apply how a drug typically would work. And this is where the modeling starts to come in. So most, but not all drugs work through reversible binding to a molecular protein target, and we call this target occupancy. And the greater the target occupancy, the greater the effect of your drug, is the sort of simple way of looking at it. So that occupancy is driven by the free concentration at the site of action, not the free concentration everywhere else, but you're hoping you understand that the free concentration everywhere else is something you can follow. And once we know what the free concentration at site of action is, we can apply two really quite simple models. Obviously, it gets a lot more complicated than this, but the two simplest ideas you would have is some drugs will appear to be exposure driven, which is this idea about the average concentration over time is, and you sometimes see it as the area under the curve. Now, this exposure-driven drug simply means then the more drug I put in, the bigger effect I will see across time. It keeps it nice and simple. Other drugs are more driven by what's called C-min, or the unbound C-min. So here is where we're trying to keep the drug above a minimum concentration for a period of time in order up to see an effect. And as long as it doesn't drop below that minimum concentration, we're expecting the effect to continue over time. Now, with this idea in our particular model, then, if we can work out the free concentration and what's happening to it, and we can combine that then with this particular model of how the drug should work, we can start to make predictions about whether our drug has an opportunity to work and be pushed forward, or whether it's going to fall down, or whether there are things we can do to, say, dosing or our dose frequency in order to change the efficacy of our particular drug. That then brings in pharmacodynamics. So pharmacokinetics is what happens to the free concentration of the drug over time. The pharmacodynamics is what happens when it gets there and is around the time. So the activity of any compound must be driven by the free concentration and how long that free concentration is there. And it's the effect of your drug based on that free concentration at the site of action which drives occupancy of your particular target. Now, life would be a lot easier if we could just find out what the free concentration was at the site of action, but almost inevitably we cannot sample there because it's normally just too invasive. So we measure the free concentration elsewhere. We apply this together within our PD model to see how that free concentration will drive an effect. Now, the simplest model you sort of get is called the Emax model. It looks basically like the Michaelis-Menten equation, if you ever sort of compare it back from there. And it simply says that there's a maximum amount of effect you could have, which is then multiplied by the concentration. And then we're dividing that by the sort of half effect that you get plus the concentration. So it's a very Michaelis-Menten-like. Now, when you apply that model, the simplest one you're going to get on a log scale, which is on our graph on the left-hand side here, you see it makes that classic sigmoidal curve that you'll always see throughout dosing. So as you increase the log concentration of your compound, you go for this sort of pseudo-linear phase, and then you start to saturate the effect of your Emax at the top of there. So if we can understand, therefore, even though the concentration of our drug is, the free concentration of our drug, is changing constantly over time, if we can link that together then to this model, we can then start to understand how, even though the concentration is constantly changing, the effect is also constantly changing as that concentration changes. But you can pull the two together and start to really understand how your drug is working and what kind of effect you expect to see on the other side. Now, this is where modelers then start to really get involved, and it's what does get a lot more complicated. And then you can then use this as part of your drug selection, the drug delivery, and how you're going to pull it together, your preclinical studies, the dose frequency you might have, and many other sort of points that we would have in there. Now, with those three ideas together, then this is an example that we then did a few years ago to a compound we managed to get to the clinic. So here is our drug, which is called CCT361814. This was optimized over a number of different rounds, as you can imagine. And then we had to be able to show in our animal model that the compound was effective. So in panel A, this is an efficacy study. So as we treated the drug once a day at 35 mg/kg as an oral dose, in the red line, you see just as the vehicle and the tumor growing. And then in the blue line, you see first of all status, the tumor fails to grow. And then we started to get a number of tumor regressions and cures of the particular mice. It starts to shrink from where you started from. All these points are the average tumors of about 10 different mice. So this particular dose, we actually was part of a design, so it was fitting with what we expected to see from our in vitro data in order to show efficacy from that. Now, a key point to PKPD is to build confidence that your mechanism of action of your drug and the efficacy that you see is linked to the protein that you believe that should be driving that effect, and at the doses and the free concentrations that make that work. So we could see efficacy at this point, and therefore the tumor is shrinking, but is that because of the way that we want it to, that it's working in the way that we think it should be working? Now, a mechanism of action of this particular drug is we had two biological pathways we were sort of looking at. One, we wanted to inhibit the heat shock response, which is sort of a key part, and we wanted a biomarker in order to understand whether the heat shock response was being switched off. And we wanted to see the unfolded protein response being switched on. So we needed a biomarker for this. And these biomarkers should be linked or correlated to the free concentration of the drug at the site of action. And how do we sort of show that? So that's when the PKPD study comes in, which is in panel B, which involves a lot more work in order to pull this together. Now, to talk you quickly through it, now what you see in the red line is the free concentration in the plasma. So this is away from the site of action, but is the place we would more regularly sample, and in a patient we'd be able to regularly sample. Now you see the highest concentration you get in the plasma occurs at about an hour. But if you look at the blue line, this is the free concentration in the tumor, which is obviously the site of action that we want because it's the tumor that we're trying to shrink. Now, the top concentration you see in the tumor is achieved after 6 hours. So there's this delay as the drug from when you've dosed it orally maximizes in the plasma, but then it takes some time to then get to the tumor, reach equilibrium with the tumor and go in and out. So this is this sort of delay that if you didn't know, if you couldn't sample the tumor, you wouldn't know this, and in the patient you wouldn't be able to see. Now we have the two biomarkers. This CHAC1 is a protein biomarker, which is of the unfolded protein response. The reason we were able to use this is because this protein has a very short half-life, and so it rapidly changes to the changing conditions of that. For the heat shock response inhibitor or inhibition of that pathway, which is HSP-A1A mRNA biomarker. Now we had to use an mRNA biomarker here because the protein that is produced from the mRNA is called HSP72, and that has a very, very long half-life of around about 70 hours. So that would mean as we switch off production of HSP72, it takes a very long time for that protein to be degraded, and therefore we would see the response. So that was no use to us as a biomarker to link together the change in the concentration of our drug to the response of our drug. So as you can see within the tumor, as the concentration increases up to this Cmax of 21 free Cmax of 21 nanomolar, this correlates with the maximum response of the biomarker, also at this 6 hour time point. But if we'd have only taken samples from the plasma, then we'd have seen this delay between these 2 points, because our maximum concentration in the plasma occurred at 1 hour, not at 6 hours. But nonetheless, when we understand that these two go together, you can see nicely a correlation as we see the significant increase in this CHAC1 protein corresponding to our Tmax. And then as the concentration in the tumor begins to drop off, then you see a drop off in the biomarker. Across our 24-hour time point period, which was our single dosing period, we could have split it, but in this particular case, we kept it as once every 24 hours. The free concentration of our compound within the tumor stays above what's called the GI50. So it's the concentration where we see half the cancer cells no longer growing when we run it from an in vitro experiment, and we stay above that for 24 hours. So this is nicely building confidence that things are working in a way that we sort of expect them to work in a dose that we're expecting them to work at. Now, if you look at then the heat shock response, which is an mRNA biomarker, where we're expecting the mRNA to go down. See, again, it corresponds to this Tmax within the tumor, not within the plasma. And then it rapidly starts to returning to baseline. It was quite difficult to get significant data with this mRNA because it's just quite a bit noisier. So we only saw significant changes then at the Tmax or the Cmax of the drug concentration. So we combined all this data together. This gave us lots of confidence that the compound was working in the biology that we predicted it should work from. And we use this then to design further experiments into preclinical toxicology, and to assess then what our therapeutic index is. We're predicting our therapeutic index to be when we go into a human in our phase 1 trial. So that thankfully means this compound is currently in phase 1 clinical trials, and we're all still really hopeful that it will continue forward into the whole process. And that is a very, very short version of PKPD.
Amazing. Thank you so much, Matt. Do we have any questions? Quick question. We have time for one question to Matt. I know there's perfect.
Oh, sorry.
I was expecting you to ask the question.
Yeah, no, I just found it really interesting, and I wish it was always that straightforward.
So I.
Yeah, I've got a project at the moment, and we haven't got the biomarker in the PD model. So we're going off purely behavioural studies. So it's been really tricky to try and calculate, you know, our PD response basically. And I wondered if you'd come over across any of that sort of project before and how you might deal with it.
In the end, you're going to have to find one, otherwise you just get stuck really. So track 1 wasn't predicted. We did an RNA-seq study to go fishing to see the things that would change. And that then gave us confidence to start looking for proteins that we hope that would change based upon that. Then you're down to something of a bit of luck that things, certainly as a protein marker, they will change quick enough that you can measure them. And so track 1 didn't make it as a clinical biomarker in the end, partly because we need 2 antibodies to do the MSD assay. The company that made one of them decided they weren't going to make that anymore, and it would have cost about 150,000 to develop it ourselves, and that was that. So you need some luck, and you need options, because they will all start to fall down as you go along, and they won't get validated. When we were doing this work within the ICR, we had a clinical biomarker team that would start to assess them for their clinical utility too. So ones that possibly worked best in the mouse possibly didn't. So you need lots and lots of options. So I think you just got to go fishing until you find something that fits, and then certainly with proteins, checking their half-lives, and can you link their half-lives and finding something that will change rather rapidly, and that's a definite advantage.